Okay, well, I first would like to start with thanking the organizers for inviting me to this beautiful place. I am from Utrecht, which is in the middle of the Netherlands. This is a picture of the city. This is the Dom Tower in the center of the city. It was already starting to be built in 1254, which is a very long time ago. And for a long time, it was one of the highest buildings in the world. Nowadays, not of course anymore. But, well, it was for a long time one of the highest buildings. Utrecht University was founded in 1636, so already a very long time ago, more than 380 years ago. And to celebrate the 380 years of existence of the university, we had a nice laser show from the campus to the tower provided by the city of Utrecht to the university. This was also in the, well, in the years that there was a lot of trade between Japan and the Netherlands, and the Netherlands was the only country that was allowed to trade with Japan, so that was a very privileged period, I would say. Okay, I'm going to talk about the plant immune system and especially the relation between probiotics that also grow together with the plant. So not probiotics in the human gut, but probiotics on the plant root and how they can stimulate plant health. But before I start, I would like to say something else. Many people who work on plants, they know about plant hormones. And one of the most important plant hormones is auxin. And auxin was discovered in Utrecht in the 30s of last century. And if you want to know where exactly it was discovered, it's on this picture. If you look on this building here, that's the laboratory where the auxin hormone, one of the first hormones of plants, was discovered by Professor Fritz Wendt. Okay, well, my first time in Sendai was 15 years ago in 2002 when I visited the Tohoku University on invitation by Hideki Takahashi and Shuhase, who is also here. Of course, we had these traditional clothing, so, well, I look much younger here, and now I'm all gray, and in those days not, but, well, time flies when you're having fun. And this was also when I visited the lab on the old campus. So this is me and this is the group. So this is Hideki Takahashi in those days, Shu. And I don't know whether there are any other people still present in the audience from this picture. No? These are the only two? Okay. And this is my group at the moment. The group is working on different aspects of the plant immune system, and I'm going to show pictures of all the people that contributed to the work, so you will see them back in the presentation. And one back. And this is the funding sources before I forget to mention them. Okay, the plant immune system was already nicely introduced by Dr. Takahashi. This is just a simplified summary of it. So it is very well investigated that plants have an innate immune system like animals have. So it all starts with a pathogen. It's recognized by its PAMPs. You get a cellular signaling leading to an immune response. But of course, successful pathogens have found ways to suppress that immune response. By injecting what we call effectors into the plant cell. They suppress the immune response and then the plant becomes more susceptible. But the plant would not have survived evolution if it would not have thought something on it. So they developed resistance genes to recognize these effectors, leading to a very strong hypersensitive response that often leads to a full resistance against the pathogen. So many of these immune responses are also regulated by hormones. So many plant hormones play a role in plant immunity, and often they are the signals that travel throughout the plant to signal other parts of the plant that are still healthy, so that they also can become more resistant against the pathogen threats. Well, the hormones that play a very important role in defense in plants are these three. So salicylic acid, oh, the wrong button. Salicylic acid, ethylene, and jasmonic acid are among the most important hormones that regulate plant immunity once a plant is infected. Nice examples are shown here. So this is just a general summary. So when a plant is attacked by a pathogen, you get local necrosis, the cells die, and then the plant starts to produce a lot of salicylic acid. This leads to triggering of a systemic immune response that we call systemic acquired resistance. And this systemic acquired resistance prepares the still healthy leaf tissue for attack against the pathogen that might come in later on. Important traits are the production of antimicrobial compounds, such as this enzyme chitinase. When the plant produces a lot of chitinase, fungi which have chitin in their cell walls cannot grow anymore, and they will be inhibited in their growth. Another example is when a pathogen is attacked by an insect. You get a chewing herbivore, and then this results in the production of a lot of jasmonic acid, the other hormone. And this jasmonic acid travels throughout the plant, and then still healthy tissue becomes alerted, and then it becomes also better protected against insect herbivory. You can see that here. For instance, then these leaves start to produce proteinase inhibitors. These are enzymes that inhibit digestion of the proteins in the insect gut, and then the insect cannot digest its food anymore. It will be inhibited in growth, and in the end it will die. And if you have a plant that produces a lot of jasmonic acid, the insects remain very small. And if you have a plant with very little jasmonic acid, the insect can grow much better, indicating the effect of jasmonic acid on defense against herbivory. Okay, there's a third type of resistance that is very important for the plant, and that is what my talk is going to be focused on. And that is a type of resistance that was discovered in Utrecht, in the Netherlands. And it is triggered by beneficial microbes that live on the roots of the plant root system. They are beneficial, they are not pathogenic, they colonize the roots, and they stimulate the immune response of the plant, leading to broad spectrum protection against different types of pathogens and even insects. And we call it induced systemic resistance, or ISR. And we have shown that the plant hormones jasmonic acid and ethylene are very important for this type of immunity. So I'm going to talk about much more on this type of resistance. Okay, so why is this an interesting type of immunity? Well, we all know that plants can grow using energy from the sun. They use sunlight to photosynthesize. They produce a lot of sugars. Those sugars are used for their growth. But up to 20% of the sugars that they produce by photosynthesis are released into the roots, via the roots into the soil. And you would say, well, that is stupid. Why would you release 20% of your energy into the soil and not use that for your own growth? But they do that in order to feed the microbial community that is around there in the roots. And they provide energy for the microbes to grow on the root system. And many of these microbes are beneficial and help the plant to grow better and to protect itself better. So what you see if you do an experiment with plants that are sterile and you grow them in soil without microbes or in soil with microbes, you will see that in the soil with the microbes, that the plant grows bigger and it has a better root system. And what you also see is that the plant is better protected against different types of pathogens. So like the probiotics that were mentioned in the previous talk in the animal and the human system, also in this case, beneficial microbes on the root system of a plant can also provide enhanced health for the plant. Okay, so in Utrecht we are very much interested in how that works. So how is the plant interacting with its microbial community in the rhizosphere? How does this lead to immunity and back? Okay. So one of the outstanding questions in this type of research is, how does a plant select the right microbes to its root system in order to benefit from it in the most efficient way. So that's also called the cry for help hypothesis. So is the plant able to recruit certain beneficial microbes to its root system when it is infected by a pathogen? So for instance, in the next couple of slides is summarized in this one. So if you have a pathogen infection on a plant, so is it then recruiting certain microbes to its root system, and what do these microbes do to the plant so that it becomes better protected? Okay, so there is a very classic example that shows that the plants can really do this. It's called disease suppressive soils. So if you grow plants in a field like this, in crop rotation, one back, if you grow your plants in crop rotation, this is wheat, and this is grown 1 year wheat, 1 year potato, the other year wheat, then potato, then wheat. And this field has a lot of, again, the wrong one. This field has a lot of takel disease. It's a fungus in the soil, and it causes this disease, and you see a lot of yellowing, and there's a decrease in the yield. Here you see a field next to it, and in this field we grow year after year wheat in monoculture. And this, in the beginning you see a lot of disease outbreak, but after a couple of years you see that it declines, and even though there's still a lot of fungus in the soil, the plants do not suffer from the disease anymore. And that is because in the rhizosphere you see a buildup of beneficial microbes on the root system, in this case Pseudomonas bacteria with a biological control capacity. They produce antibiotics that inhibit growth of the fungus in the soil. So in this case it's a nice demonstration of the fact that the plant is able to recruit certain microbes to its root system that protects it against a fungus, in this case the takel disease in wheat. We use this example to investigate this in more detail. So we did the same experiment with Arabidopsis grown in soil, in a natural soil on which a lot of Arabidopsis is growing in nature. So what we did, we grew Arabidopsis on that soil, and we did different treatments of the Arabidopsis plants. So we either had control plants that were not treated at all, and we put them on the soil. We had plants that were inoculated with botrytis, plants that were inoculated with a downy mildew pathogen, or plants that were treated with jasmonic acid or salicylic acid. And then we monitored the composition of the microbial community on the roots after those treatments. Yeah? And what you then already can clearly see is that if you look at the microbial diversity in the unplanted soil, a few centimeters away from the roots, so it doesn't contain any roots, you see that the microbial community is much different from the microbial community on the root system of the plant. So it clearly shows that the plant is able to change the microbial community around in its rhizosphere in comparison to the unplanted soil. Yeah? But what you also see is that the different treatments have different effects on the microbial composition. And that becomes more clear on this picture. Here again, we look at the OTUs, and an OTU means operational taxonomic unit. So it can be a species or a genus, but so you have to look at it as a well, let's say a microbial species. And if you look at microbial species, you will see that there are 3 species standing out in this experiment: 106, 97, and 107. And if you look in more detail, then you see that these microbial species increase specifically in response to infection of the plant by the downy mildew pathogen Hyaloperonospora arabidopsidis. So this is after 1 week, this is after 2 weeks. You will see that this species, 97, which is a Stenotrophomonas, it significantly increases in the rhizosphere already after 1 week, and after 2 weeks it's much higher. This species as well, it's a Xanthomonas species. After 1 week, it's already significantly higher, but after 2 weeks, even more, and this one as well. So these 3 species apparently accumulate in the rhizosphere on roots of plants that have been infected by the downy mildew pathogen in response to the infection. Okay, so what happens when a plant becomes colonized by certain microbes? What are the processes that then become engaged? So this is a schematic overview of that. So here we have a plant root. This is the outside of a plant root. This is, these are the exudates of the roots. So these are the photosynthetes or other molecules that are excreted by the roots. They are recognized by the microbes, and when the microbes react on it, they start to colonize the roots, and the first thing they need to do is make, how do you call that? Biofilm, right? They form a biofilm on the roots, and if they form a biofilm, they are with many microbes on one place, and then they start to activate certain activities like producing antibiotics and stuff like that. But not only that happens, you also see local reactions in the roots and also systemic reactions in the plant in response to this colonization. Well, the first thing that we tested with these strains was what is the effect on biofilm formation. So this assay, if you look at the colors, the more purple, the better biofilm is formed. So if you use only the single strains that accumulated in the roots, on the roots, you see that they can form a biofilm, but the biofilm is much stronger when you have them in combination, right? So these three microbes that start to accumulate in the rhizosphere after infection with the downy mildew pathogen, they are together capable of forming a much stronger biofilm than either of them alone. So apparently that is something that happens in the rhizosphere. Upon infection, these three become recruited by enhancing the biofilm formation. So when they form a biofilm, then of course they are with a lot of microbes together, and then they start to activate certain activities. Here's one example of that. This enhanced biofilm formation can be based on the fact that they stimulate each other's growth. So this is an assay where we looked at the effect of one microbe on the other. So this is 106, 107, and 97. So if you combine X and M, so that's X is this one, M is that one. If you combine them together, you will see that M is stimulated by the presence of X, you see? And also here you see that S is growing much better when it is close to X. So it nicely shows that if you grow these microbes together, they grow better, leading to an enhanced biofilm formation. So what do these microbes then do when they are together? So if you apply them together on the root system, so X, S, and M is the combination of the three microbes on the root system. If you look at the plants and you grow them sterile, so without microbes, they grow nicely on a plate. But if you apply the microbes to the root system, you see that they form much more lateral roots. They form more side roots, which means that the root system becomes bigger, so it is able to take up more water, more nutrients, and then grow better, leading to an enhanced fresh weight of the shoot. So the plant grows better and produces more fresh weight than without the microbes. Also, if you do that in soil, you can see the effect. So this is the control situation. These are plants grown in sterile soil, and these are plants grown in soil through which these three microbes have been mixed. And then you see that the plants are clearly bigger than on the left side. Okay, well, this is typically an auxin effect. So I mentioned already on my first slide auxin. That is an important growth hormone of plants. And what these microbes do, they stimulate the auxin response in the roots. And we know that because we have a marker for the auxin response. In this case, it's a yellow fluorescent protein fused to an auxin promoter. And then you see, if you apply the bacteria to the roots, you see a stronger auxin response than in the roots without the microbes on it. We have done a lot of experiments also with mutants. It appears that these microbes stimulate the auxin response in the roots. This leads to more root branching, and not only root branching, also root hairs are longer. On the left-hand side, you see a root without bacteria, and on the right-hand side, the root with bacteria. And you see that the roots are much longer, also enhancing the surface area of the roots so that it can take up more water, more nutrients, and grow better, leading to enhanced shoot fresh weight. Okay, well, does it protect the plant against infection? Well, the answer to that question is yes. So if you apply the 3 microbes to the plant, like here, so here we have the Arabidopsis plant, we grow them in the presence or the absence of the 3 isolated microbes, and then we inoculate again with the downy mildew pathogen, the pathogen that initially recruited these microbes to the rhizosphere. And then you nicely see that without the 3 microbes, you get a certain level of infection. But if you grow the plants in the presence of these microbes, you see that the level of infection is significantly decreased. So apparently, what we can conclude from this is that after infection, the plant recruits certain microbes in its rhizosphere. These microbes stimulate plant growth, and they also protect the plant against the pathogen that originally initiated their recruitment. Okay, well, this type of resistance that we observe is called induced systemic resistance. And in the past, we have performed a lot of research on this type of resistance. Here you see an example. On the left-hand side, you see a plant that is inoculated with Fusarium oxysporum, an important pathogen. On the right-hand side, this plant was similarly infected, but this plant was grown in the presence of the Pseudomonas bacteria, one of a group of bacteria that's very well able to induce systemic resistance in plants. And this leads to protection not only against Fusarium, but against many types of pathogens, including Pseudomonas syringae, Erwinia carotovera, Botrytis cinerea. So it provides a broad spectrum resistance against many types of pathogens. Well, in the past, we have published a lot on this type of resistance, so I'm not going into much detail, but the bottom line is shown on this slide. So the roots become colonized by the rhizobacteria. This leads to the production of a signal that is then spread throughout the plant. The signal is not known yet. And this signal then leads to priming of the above ground plant tissue so that these tissues become in an enhanced level of alert so that they respond quicker to pathogen infection than non-primed plants. An example of this is shown here. So here you have a control plant, and this is a defense marker gene. So after infection, a plant that is grown without the rhizobacteria in the soil responds to pathogen infection, as you can see here. But if you have a plant that is grown with the rhizobacteria on the roots, you see that this defense marker gene is activated earlier and to a stronger level than in the control situation. And this is a nice example of priming, which means that the plant is showing an accelerated defense response in comparison to the control situation. So we like to visualize that like this. So if you have a plant that is colonized by the rhizobacteria, you get a stronger plant, grows better, and is defense primed, so it's better protected against all kinds of pathogens. Well, in the last part of my talk, I would like to zoom in to what is happening in the rhizosphere in early stages of the onset of induced systemic resistance. And so what is happening in the roots when these roots become colonized by these ISR-inducing rhizobacteria? Well, of course, we first started to look at gene expression. If you look in the shoots, you don't see a lot of changes in gene expression in the absence of a pathogen. But in the roots, a lot of things are happening. I will not go into too much detail, but one of the most important genes that change in the roots is a transcription factor gene, so a gene that regulates other genes. It's called MIP72. It's a transcription factor gene that is only expressed in the roots, not in the shoots. And it's specifically expressed in response to bacteria that can stimulate this systemic immunity. So we have our pet strain, Pseudomonas simiae WCH417. It induces MIP72 very strongly. Another Pseudomonas biocontrol bacterium, WCS358, also induces this gene, but a Pseudomonas fluorescens that does not induce resistance in Arabidopsis. It also does not induce MIP72. So there's a clear correlation between induction of MIP72 and the onset of systemic resistance. And if you have a mutant of the MIP72 gene, it does not develop this systemic immunity anymore. So it's really a gene that is required for the systemic immunity. Here you see expression of the gene in the root of Arabidopsis. So on the left, here you see an Arabidopsis root colonized by the biological control strain, which is labeled with YFP. So yellow are the bacteria, red is the root. And here you see the root of a reporter line for MIP72, MIP72 promoter and GFP reporter gene. So you see without the bacteria, there's very low expression only in the vascular bundle. But if you apply the bacteria to the roots, you see a very strong expression of the MIP72 gene, especially in the epidermal cells of the roots. Well, here you can have a look at the whole root system where it is expressed, and it's especially expressed in the maturation zone of the roots, of the lateral roots. Well, it's a transcription factor gene, so it regulates other genes, and one of the most important other genes that it regulates is a gene with the name BGLU42. It is a beta-glucosidase, and the beta-glucosidase, what it does, it is cleaves off the glucose molecule of a secondary metabolite. So for instance, abscisic acid can be glucosylated, and if you have beta-glucosidase activity, you can cleave off that glucoside, and then you get an active molecule. So it is an enzyme, and it is regulated by MIP72. It is very important because if you overexpress BGLU42, you get constitutive induced systemic resistance. So if you have a transgenic plant that is overexpressing BGLU42, you get enhanced level of resistance in the plant. So it's a very important gene for ISR. And if you have a mutant that is not able to produce BGLU42 anymore, you don't get any ISR anymore. So it's a very important step in the onset of systemic resistance. And if you look at where this, these are the data to support it. So here we have BGLU42 overexpressor, 3 independent lines. It is enhanced resistant against botrytis, enhanced resistant against a bacterial pathogen, Pseudomonas syringae, and enhanced resistant against the downy mildew pathogen. So it provides broad spectrum resistance that is typical for ISR. Okay, well, another thing that is known about MIP72 is the fact that it is also induced in response to iron deficiency. So when plants grow in soil with very low iron, you also see that this MIP72 gene is activated. So the only 2 ways by which MIP72 can be activated is by these beneficial rhizobacteria and by conditions of low iron. So if you, we know that because that's another field where a lot of people have studied this. So if you look at the overlap between genes that are activated in response to iron deficiency and to colonization of the roots by the beneficial microbes, you see a lot of overlap in genes. So, and there are a couple of very important genes that relate to this iron uptake response, and those are FRO2 and IRT1. I will show later what they do. But they are also both co-regulated with MIP72. So when MIP72 is activated by the 2 ISR-inducing microbes, FRO2 and IRT1 are also activated by these beneficial microbes. Just to give a summary about what is this iron uptake response doing, so whenever a plant senses low iron in the soil, and many soils have a very low iron, then they start to respond to that so that they can take up more iron from the soil. And what happens is that the plant starts to excrete protons so that you get lower pH of the soil. This leads to enhanced solubility of iron in the soil. And then you get, this iron is then reduced so that you get iron that can be taken up by IRT1, and then the plant can grow better because it takes up more iron. What is MIP72 doing in this response? Well, MIP72 is also activated, and what MIP72 does is, now we need to do the MIP72 is very important for the production of certain phenolic compounds. When this system is activated by low iron or by the beneficial microbes, then the plant starts to produce certain phenolic compounds, and these phenolic compounds are then produced inside the root, and the BGLU42 activity is required in order to excrete them into the rhizosphere. And when they are excreted into the rhizosphere, they also help to increase the solubility of the iron, and the plant can take up more iron. Yeah? Here you can see that on the slides. So on the left-hand side, you see a wild-type plant, and it is producing a lot of these phenolic compounds. We also call them coumarins. And these coumarins help to take up iron so that the plant can grow better and you get growth promotion. And the MIP72 mutant is not able to produce these phenolic compounds at all. So MIP72 is very important for these coumarin production. BGLU42, the mutant that is very important for this ISR response and which is regulated by MIP72, it is still producing these coumarins, but they are not excreted into the rhizosphere. And we know that because if you grow your plants in wells like this, and you take out the plants, then you still see that the coumarins are excreted because they remain into the medium, but MIP72 doesn't produce them at all, and BGLU42, it's not in the medium because it remains in the roots, indicating that this BGLU is very important for excretion of these phenolic compounds into the rhizosphere. So from this we can conclude that MIP72 is a very important regulator of coumarin production, and BGLU42 is very important for coumarin excretion. What I forgot to mention is that the activity of MIP72 is regulated by volatiles that are produced by the microbes. Because if you only apply volatiles of the microbes to the root system, you see activation of MIP72 and production of these coumarins. We know that because if you use a system like this, where you grow the plants on this side of the plate, and on the other side of the plate you apply the bacteria, and this is physically separated, then and only the volatiles can go from here to there, then you get activation of MIP72, and this is how MIP72 is activated in response to low iron. So the volatiles of the bacteria are responsible for the activation of the MIP72. Okay, so to summarize this, and then I go to the last part to relate it to the microbiome. So what we know now is that the beneficial microbes, they have volatiles. These volatiles activate MIP72 and the whole iron uptake system in the roots. This MIP72 is important for the production of these phenolic compounds. These phenolic compounds lead to, they remain inside of the root unless you have BGLU42 activity. When you have BGLU42 activity, the glucoside molecules are cleaved off these secondary metabolites, and then they are secreted into the rhizosphere. And what we also know is that if you overexpress BGLU42, you get systemic resistance. So what we now think, and that is also something that the Sietske, who was also here in the audience, worked on, this BGLU42 activity is possibly also important for generation of a systemic signal, possibly one of these phenolic compounds, that is then traveling to the upper part of the plants, leading to systemic immunity. But then the question is, of course, why do these bacteria stimulate this response? What is their benefit? So what we think is that these phenolic compounds, many of them have antimicrobial activity. They are plant antibiotics, and they are excreted into the rhizosphere, and they will have an effect on the composition of the microbial community. So they will inhibit growth of certain microbes and not of other microbes. And that's something that we wanted to know. So do these ISR-inducing microbes induce antibiotic production by the plant in order to benefit from it, because it is possible that these phenolic compounds inhibit growth of other microbes that do not induce ISR. That's something that we tested. I just wanted to show Sietske. She's in the audience, and when she was in our lab, she worked on testing the role of BGLU42 in the production of this ISR signal, but we're still working on it at the moment. So in order to know what is the effect of these phenolic compounds on the microbial community, we first needed to identify these phenolic compounds. And that was done by Janis in the lab. So he grew up wild-type plants, BGLU42 plants and MIP72 plants. And you see that the wild-type plants produce a lot of these phenolic compounds. BGLU42 produces them, but they stay inside the roots. And MIP72 does not produce them at all. And we did a metabolite profiling of the exudates and of the root interior to identify the phenolic compounds that are produced in a MIP72 dependent manner. And the bottom line is that the most important phenolic compound that is produced by the roots in a MIP72 dependent manner induced by these rhizobacteria is scopolamine. So scopolamine is a precursor of scopolamine, and scopolamine is produced inside the roots, and scopolamine is excreted into the rhizosphere. So scopolamine has the glucose molecule, and when that's cleaved off by BGLU42, you get scopolamine, and it's excreted into the rhizosphere. That's the bottom line of this analysis. And then we wanted to know what is the activity of this scopolamine on the different microbial communities. So first, Janis tested, and I think Sietske contributed on that as well. I should have put your picture there as well. Tested the effect of scopolamine on this fungus, which is a fungal pathogen on plants, Fusarium oxysporum, and Verticillium dahliae, which is another root pathogen. And you see that both root pathogens are very sensitive to scopolamine. So you can imagine that when the plant starts to produce scopolamine into the rhizosphere, this reduces growth of these fungal pathogens. However, if you look at the beneficial microbes that stimulate the production of scopolamine, like WCS417 and WCS358, the two ISR inducers, they are not sensitive at all to this scopolamine. So you can imagine that when the plant starts to produce this phenolic compound, it will benefit the ISR inducers over the pathogens into the rhizosphere. So to analyze that in more detail, we did a microbiome analysis of plants that were grown in soil, the wild type and the MIP72 mutant, to see what is the effect of MIP72 activity on the buildup of the microbial communities in the rhizosphere. So here you see the results of a microbiome analysis. So again, we looked at, we isolated all the microbes from the root systems. We isolated the DNA. We did shotgun sequencing of the whole microbial community. And we checked the differences between wild-type Columbia plants and mutant MIP72 plants in terms of microbial diversity. And again, you see that the unplanted soil is much different from the root environment, but there's also a clear difference between the wild type and the mutant in terms of microbial diversity. So apparently, MIP72 activity in the wild type has an effect on the microbial diversity in the rhizosphere, because the MIP72 community is much different from the wild type microbial community. And we also, because we did shotgun sequencing, we had an opportunity to look at all the different species that were present in the rhizosphere of the wild type plants and of the MIP72 mutant plants. And then you can identify which bacterial species and fungal species are sensitive to MIP72 activity, so sensitive to this scopolamine. And then here you see the results of that. So on the, here you see all the microbial species that are negatively affected by MIP72 activity. And here you see all the microbial species that are positively affected by MIP72 activity. And they clearly come from different phyla. So apparently MIP72 has a large effect on the composition of the microbial community in the rhizosphere. And if you look in detail, you also see that of the genus Pseudomonas, there are Pseudomonas species that are negatively affected by MIP72 activity and Pseudomonas species that are positively affected by MIP72 activity. So what we now are interested in is, well, does this population of microbes, is that enriched for microbes that induce systemic immunity, while the ones that are here do not induce a systemic immunity. So that provides more insight into how a plant is able to recruit the best microbes to its rhizosphere so that it enhances the chance that it has protective microbes in its rhizosphere over, well, the other option. So this is the last slide. It summarizes what I talked about. So I talked about in the beginning about the fact that if a plant is infected by, for instance, the downy mildew pathogen, it starts to recruit beneficial microbes. The three microbes that I mentioned, they form a better biofilm and they stimulate growth, they stimulate immunity, leading to better protection. And I show that, yeah, so that you have a better protection against the pathogen that originally started to recruit them. And I showed some information about how this works at the level of the roots. So in the roots you see, oh no, that's the wrong one. In the roots you see that the beneficial microbes stimulate MIP72 activity. This MIP72 activity is responsible for the production of phenolic compounds. We identified scopolamine as one of the most important phenolic compounds. By BGLU42 activity, it is excreted into the rhizosphere. In the rhizosphere, this phenolic compound is important for the selection of beneficial microbes. Some are enhanced and some are suppressed. And what we are now working on is to see, well, can we identify a phenolic compound which is a candidate for the ISR signal that is then responsible for the systemic immunity that we observe. So why is this all important? Well, it is important because we like to see that the agricultural crops that we have at the moment, they are a little bit obese. They are dependent on fertilizers, they are dependent on pesticides. Of course, they produce a lot then, but they also are very sensitive. And what we would like to, so we see it like this. So if you have the modern crop at the moment, the modern crop lost a lot of traits that the wild species still have. So many of these traits are most likely related to its ability to maximize the profitable functions from the root microbiome. So like the yellow and the red traits, they relate to, well, beneficial functions from the root microbiome. They are lost in the modern crops because they are never selected for. And what we like to do is investigate what are these yellow and these red traits and what are these beneficial microbes that provide these beneficial traits, and how can we bring that back into the future crops so that we can grow our crops with less fertilizers and with less pesticides and still provide a healthy crop for agriculture. Well, for the Dutch economy, it's very important. Maybe you don't know it here in Japan, but in the Netherlands, if you look worldwide, all the vegetable seeds that are produced come from Dutch breeding companies. All the vegetables, every seed that is planted in the soil every year, 40% of all those seeds come from Dutch plant breeding companies. Also seed potatoes, so the potato that you put in the soil to produce more potatoes, 60% come from the Netherlands, and also 45% of all the starting materials like flowers and bulbs also come from the Netherlands. So for the Dutch economy, it's very important that we find better solutions for using less pesticides and growing more healthy and sustainable crops. And with that, I would like to say domo arigato. And this is the picture of last night.